Contextual control of operant behavior of a North American zebrafish. Contextual control of environmental events on the operant responding of a North American zebrafish was demonstrated in the current experiment. Baseline results demonstrated that two discriminative stimuli, a blue light and a red light, failed to produce differential responding across the contextual conditions when reinforcement was not provided contingent upon the operant responses. In the training phase, reinforcement in the form of fish food was provided differentially for two target operant behaviors, swimming left and swimming right, in each of the contextual conditions. Continuous reinforcement schedules were identical across the conditions. Swimming right was reinforced under the blue light condition, and swimming left was reinforced under the red light condition. Results showed that when differential reinforcement was introduced, the colored lights effectively demonstrated contextual control over the fish's swimming behavior. Results in discussion are provided from a contextual behavioral perspective on animal behavior. Differential reinforcement has proven to be effective in countless studies dating back to the beginning of behavior analysis, when B.F. Skinner used it to shape behavior of hungry pigeons. Presently, differential reinforcement is widely used in the field, particularly with people with developmental disabilities. This is apparent from the extensive number of reports that have been published in recent years, including studies involving tact acquisition, increasing eye contact, increasing food repertoires, and decreasing elopement, and the list goes on. One factor that many differential reinforcement studies have not addressed, however, is contextual control, or the influence that contextual conditions can have on behavior. This is most likely due to the fact that concepts such as contextualism and relational responding are not generally accepted by devout Skinnerians. It has been noted that there hasn't been a single article published in Behavior Analytic Journal that argues the existence or logic of relational operants. Rather, traditional behavior analytic literature simply avoids engaging in such issues. The present study contributes to this research by examining the contextual control of environmental events over the swimming behavior of a non-human subject, a North American zebrafish. Zebrafish are said to have a variety of characteristics, including their short maturation and capability of complex behavior, which make them an effective complement to classic mammalian models. Additionally, the retina of the zebrafish have many structural similarities to human retina, making them valuable subjects for the present study, which involves the visual system. Using different colored light to create different contextual conditions, this study will examine whether the zebrafish's behavior can be contextually controlled or not. The results of this study will foster opportunities for future translational research between basic and applied settings, which could lead to the development of new contextual behavioral technologies for human subjects in applied settings. The current study involved a single North American zebrafish. The subject was female. The subject resided in the same tank used in the current experiment and remained on a 12-hour light-dark cycle in which she was kept in light for 12 hours and kept in dark for 12 hours each day. They were overhead fluorescent lights and were kept on at various times daily. The subject was fed fish flakes daily, and previously the subject participated in light discrimination studies and hoop training experiments. The experiment was conducted in a large laboratory setting from approximately 7:30 to 9:30 p.m. Researchers conducted the experiment on a concrete floor close to an outlet. The procedure consisted of a multi-element design with baseline training and maintenance phases. There were two contextual conditions in which the subject was trained to exhibit two different operant responses and a control condition. In the blue light condition, the response trained was subject moving to the right side of the tank by passing the tape with at least half of its body and remaining inside the parameter for at least 2 seconds. In the red condition, the response trained was the subject moving to the left side of the tank by passing the tape with at least half of its body and remaining inside the parameter for at least 2 seconds. The experiment included a control condition to ensure that the behaviors occurred in the discriminated conditions. In this condition, a response was recorded if the subject exhibited either of the responses. Experimenters measured the percentage of correct responding performed independently within the condition. For the training condition, experimenters additionally recorded their percent value of the prompt needed during each trial. For baseline, each condition was presented in 3 trial blocks on an alternating schedule, and experimenters recorded whether a correct response had occurred. No reinforcement was administered during this phase. Once steady rates of responding occurred, experimenters began the training phase. For the training phase, each condition was presented alternatively in 5 trial blocks. The training condition included a prompting procedure to ensure correct responding. Experimenters placed lines in the apparatus to represent 25, 50, and 100% of the space in the apparatus that was considered incorrect responding. After 5 seconds of incorrect responding, one experimenter placed her hand in the apparatus at the opposite end of the correct response for the trial. Then the experimenter moved her hand to the next line every additional 2 seconds of incorrect responding until a correct response occurred. For the maintenance phase, experimenters only included the red light and blue light conditions. Everything else was the same as the baseline phase. They calculated IOA data for 33% of trials for every third trial by recording results simultaneously and independently for those trials. Agreements were divided by disagreements and multiplied by 100. The IOA for the baseline phase was 100%, 92% for the training phase, and 100% for the maintenance phase. A third researcher calculated procedural reliability for 33% of the trials by observing the experiment and calculating the percentage of steps done correctly by the experimenters. The present study demonstrated experimental control by using a multi-element design with a control condition. It can be seen in the graphs included that during the training condition, the zebrafish learned to correctly discriminate between conditions. However, correct responding of swimming to either side for 2 seconds remained low for the control condition. The current study has added to the research by showing that zebrafish's behavior can in fact be contextually controlled by using lights. Further research will be necessary to address and control for the experiment's limitations. Food as reinforcement had its limitations due to the difficulty to deliver the flakes and the fish becoming satiated. Therefore, another reinforcement contingency was occurring. It is possible that the removal of the lights and prompts could have served as negative reinforcement for the target behavior of the fish. Also, the prompting procedure was done with the experimenter's hand, allowing for subjective judgment to occur. Future research should control for biases like this with increased procedural fidelity. Sessions and trials were short in duration in order to fit time constraints. Therefore, longer trials should be explored. Finally, keeping track of the time during each trial was found to be challenging with only 2 researchers. A third researcher would have proven to be useful in this regard. Introducing new contextual conditions other than the visual colored lights for which the zebrafish would have to discriminate between could expand on this present study.